Hello and welcome to the PyMol tutorial. In this video, we are going to learn how to calculate the torsion angles of proteins by using PyMol. In previous video, we have seen what the torsion angles in a protein molecule are and how they dictate the protein structure. You can watch the video if you haven't watched it already, and the link is also available in the description. In this video, we are going to calculate the torsion angles by using three different types of examples. First, we build a peptide chain in the PyMol and then calculate the torsion angles. Second, we download a PDB file and see how to calculate torsion angles for an alpha helix. And finally, we calculate the dihedral angle of a cis peptide. During this tutorial, I will also explain some beautiful functions of proteins which I am going to use in this tutorial. So let's start without any delay. To calculate a dihedral angle in proteins, we need 4 atoms or 3 consecutive bonds. Keep the bond in the middle whose rotation angle you are going to measure, and take one more atom on each side. Let's assume these 4 atoms as ABCD, where bond BC rotation angle is being calculated. Now look through the bond BC by keeping atom B towards yourself. Here, bond AB is the nearest one and the bond CD is the farthest one. And finally, measure the angle of rotation around the bond BC via AB to CD. If the rotation is clockwise, then it will be a positive number from 0 degrees to 180 degrees. If the rotation is anti-clockwise, then it will be a negative number from 0 degrees to minus 180 degrees. Let's begin first with the PyMol builder. Open a PyMol session and go to the builder, which is located on the top right menu. In the pop-up window, navigate to the protein tab, then click on alanine residue. Now select the option Create as New Object. So it will create an alanine residue. Now you may notice the sphere sizes are too big, so let's change them. Go to Settings, then Edit All. Type sphere, then change the value for sphere scale to 0.25. Now zoom in and click on the carbonyl carbon to add upcoming alanine residues. A tripeptide with 2 peptide bonds will be good enough for us to work. Close the window and click Done. Now hide the cartoon, show spheres, and finally remove the hydrogens for ease of representation. So the tripeptide is ready to work. Let us begin with the torsion angle omega. Look at this peptide bond and try to look through this peptide bond to understand the angle of rotation, that is omega. Now go to Wizard under the main menu and select the measurement and choose the option dihedrals from the measurement mode. And now follow the instructions on the top left corner or simply click on 4 consecutive atoms. We need to keep the peptide bond in the middle and C alpha carbon atoms on both ends. So let's do it. 1, 2, 3, and 4. This is the torsion angle omega, and it is perfectly 180 degrees. Let's move on to the next peptide bond. Try to understand the peptide bond and then the angle of rotation. Similarly, click on 4 consecutive atoms 1, 2, 3, and 4. I'm changing the color to cyan to match with our color code and hiding them for the moment. Let's move on to the next torsion angle phi. Find the bond N2C alpha. Look through this bond to understand the torsion angle and proceed with the measurement as previous one. 1, 2, 3, and 4. Always we should look from N terminus to the C terminal side to assign the sign of positive if it is clockwise rotation and negative if it is anti-clockwise rotation. Repeat the process for the second phi angle in the same way. We look here from C terminal side, and that's why the signs are reversed, which means the clockwise rotation has a negative value. Now let's calculate the torsion angle psi. This is the rotation angle around the bond C to C alpha. Look through this bond and understand the rotation around this bond and calculate the torsion angle by clicking the 4 consecutive atoms by keeping the C to C alpha in the middle. Continue with the next psi angle in this tripeptide. One, 2, 3, and 4. Let's change the color of torsion angle psi to magenta to match our color code. Let's see all 3 torsion angles together: omega in cyan, phi in yellow, and psi in magenta. Now let's move on to the second part. In this part, we will download a PDB from Protein Data Bank and calculate the torsion angles. This is the structure of Thioredoxin, an antioxidant enzyme that is present in almost all organisms from archaea to mammals. Before we proceed, let's have a small description about this enzyme's function. The thiol groups of cysteine residues in proteins tend to form disulfide bonds. These unwanted disulfide bonds must be reduced for a protein to function properly. Thioredoxin is one of such proteins that keeps cysteine residues in reduced state. Thioredoxin acts on large number of substrates. Barley alpha amylase substilin inhibitor or BASE is one of them. The germinating seeds of barley contains amylase, which degrades starch for energy source. BASE inhibits amylase and avoid premature sprouting by preventing starch hydrolysis. 2 cysteine residues, 143 and 148 in BASE, forms a disulfide bond, and they are the target site for Thioredoxin. Thioredoxin contains a highly conserved CGPC motif. The cysteine residues of this CGPC motif transfers the disulfide bond from substrate to thioredoxin itself. The N-terminal cysteine of CGPC motif makes a nucleophilic attack on the substrate disulfide bond, which results in an intermediate disulfide bond between the CGPC motif and the substrate. The C-terminal cysteine of the CGPC motif further makes a second nucleophilic attack on this intermediate disulfide bond and results in the final product. The oxidized thioredoxin further reduced by another enzyme called thioredoxin reductase by spending an NADPH. Let's see how to calculate torsion angles from this PDB. By clicking on S, you can display the sequence. I am selecting a short alpha helix here and show as sticks, color by element, and then hide the rest of the cartoon and show the selected helix as spheres to make it look more clear. Now let's zoom in. Look at the top view of an alpha helix. Now look at this small stretch and identify three torsion angles: psi, phi, and omega. So we are ready. Let's go to Wizard and select the option Measurement. Then again select the option Dihedrals from the drop-down menu on the right-hand side. Now look at the peptide bond and select the 4 consecutive atoms by keeping the peptide bond in the middle: 1, 2, 3, and 4. And I am changing the color to cyan to match with our color code. Now let's go to phi and select the 4 consecutive atoms: 1, 2, 3, and 4. This is the torsion angle phi and it is -63.4 degrees. And finally, let's calculate the torsion angle psi. One, 2, 3, and 4. Let's change the color of psi to magenta. And it is -49.5 degrees. Think about the Ramachandran plot while you are calculating these torsion angles, and imagine where these values are falling under. Let's move on to the final section, calculating dihedral angle from a cis peptide. In this part, we will use an example of alpha L-arabinofurantosidase, a glycoside hydrolyzing enzyme. This enzyme has a catalytic domain and an arabinose binding domain. The catalytic domain has a cis peptide formed by 2 cysteine residues. This cis peptide recognizes the arabinofuranose and helps in the catalysis. Let's have a look at this cis peptide and also observe the orientation of peptide planes. This is the cis peptide. Go to Wizard, Measurement, and Dihedrals. 1, 2, 3, and 4. Interesting. From 180 degrees in trans configuration to 0 degrees in cis configuration, how the torsion angle omega has changed the peptide backbone. So this is the end of this tutorial, and thank you very much for watching this video. Like this video and comment below if you have any questions. And please do subscribe the channel. See you in next interesting video. Until then, take care. Bye bye.